let's talk about a ring species. Because I'm going to get to, eventually, there was no Adam and Eve, historically. Philosophically, psychologically, theologically, oh yeah, sure. But species do not have founding members. It's always a population. Always a population. So let's illustrate that. How many have heard of the concept of a ring species? Okay, there's a biology major here. A ring species. It's where you have a collection of populations, and then there's a physical break. In California, around the San Joaquin Valley, there's this desert area here where these poor salamanders can't go visiting. And what you'll see is a continuous gradient, breeding gradient, where this breeds with that, and that with this, and this with that. And by the time you get all the way around the ring, the coloration is completely different, where this goes, I'm not going to date that. That's a whole different thing. Probably not even legal in Florida. So, is that a different species from that? What's the definition of a species? As you may imagine, it's all made up. It's two groups of organisms that cannot interbreed. That's it. That's all it is. That's pretty artificial, right? You could force these guys and now they're a species suddenly. It's totally made up what a species is. Let's go a little further here. Of course, if you do well in this course, you won't do well in any of your other biology courses. Okay, let's talk about another ring species, a classic example, the gull. You start here in England. This is Norway. And you go Greenland, North American herring, Alaska. Is that Alaska? Whatever that is, and back to here. That's Siberia, right? Okay, yeah, got it. These do not mate with these. They don't even recognize them as gulls. Like, what are you? I don't know, what are you? No way. Forget about it. No way. I go to a movie by myself. Ring species, where you have a gradient of populations that exchange genetic information, but this one would not ever breed with that one. By definition, they are different species, but yet they are connected by a continuous chain of mating possibilities. So let's take that concept of a ring species and take that ring and straighten it out. Now we have a linear gradient, right? And we take that and then we shove it back down through time. And you see that we are the result of a continuous series of interbreeding populations. Would we interbreed now with Homo erectus? Well, most of you, the guy on the back does. I don't know, give me a number. But every species is a continuous series through time of interbreeding populations. There's no one point where you can say, aha, this species is different from that species. There are populations that are interbreeding, and then they undergo different selective pressures, and then they can no longer interbreed with each other. That's how species happen. But there's no founding one member. The question, which came first, the chicken or the egg, is not the right question. There's always a diversity. Someone asked me, they're going, well, human evolution, well, if that's true, why are we all the same? I went, dude, look around you. We are in dynamic flux as a species. We are a very recent addition. More about that, you'll see. When did Homo sapiens become a species? That's also part of this, but we have a few more detours. What about the origin of eukaryotes? And we've alluded to this, the fact that many of the structures that eukaryotes have, they got it from endosymbiosis. Cilia that you find in your, I guess, esophagus. Spirochetes, gametes, flagella. The nucleus itself came from Mama Archaea. You know, how many have heard of, the bacterial RNA polymerase enzyme that reads the DNA and makes the RNA? You've heard of the 4 subunits, beta, beta prime, a couple of alphas, and omega nobody knows about, sigma, get the ring. We don't have that. But what we do have is from another unicellular organism, a whole nother kingdom called Archaea. The polymerase that we have that reads our genome has 12 subunits. This little Archaeobacter that lives in the oceans and in hot springs and all over the place, it has those same 12 subunits. In fact, that's where we got them. These Archaea invented histones. The Archaea invented all the major proteins that recognize what a gene is. Our nucleus came from Archaeobacter, probably our cytoplasm from eubacteria. We are a composite, a chimera. Things are not as simple as they seem. This particular protein, TBP, Tata binding protein, that structure is billions of years old. This is what you find. This is how Archaea recognizes where the beginning of a gene is. There's a sequence that says T-A-T-A-A-A, Tata. Except Archaea reads it backwards, where it's ta. But for us, it's Tata. That ability to recognize the minor groove, that's crazy. Minor groove, proteins don't recognize minor groove, it's major groove. This thing that we have in our nucleus to recognize where genes start is the same TBP. You could superimpose them in terms of structure and you wouldn't be able to tell the difference. Highly conserved. We inherited it from Archaeobacter. Oh man. And this is the other transcription factors that you'll learn about. It looks like just a big blob. It sort of looks like that statue in front of Turlington, doesn't it? Now you finally know what that thing is. What is this about? This is Mars, right? That's where we're headed. The first eukaryote on Mars is going to be a little plant called a Arabidopsis. Anyway, that's what it looks like right now today. What? I've already interrupted my own lecture? Okay. Now we change the pace. And we talk about us as an organism. What's the time? 5 minutes? Let us plow on further into this fun. Well, what we got here? Australopithecine. Oh, yeah. Lucy, oh yeah, this guy. Australopithecus afarensis. She was a baby doll. Not considered a member of the genus Homo. But that was 3.2 million years ago. When did we split from the chimp line? about 5 to 7 million years ago. 5.5. Speak for yourself. No, that's, yeah, the estimates vary and depends on what you, but that's the ballpark, 5.5 million years ago. And this is the beginnings of that line. So let's get down and finally talk about some molecular biology. I'm going to talk about one gene, SIRGAP2. This gene has been put in mice. The version that's in Homo sapien has been put in mice to see whether they get smart. Let's see what it does. SIRGAP2 has been duplicated in Homo sapien. Not so much in other mammals, but in us, it's been duplicated. And when it duplicated, it messed itself up. It wasn't the full-length gene that got duplicated. It was mutated and truncated and shortened. And because it was shortened, it couldn't function properly. So when it still had affinity for the wild type SIRGAP2, and the mutant form of SIRGAP2, which is in us, and the original, the mutant form binds to our original and makes our original SIRGAP2 not work as well. SIRGAP2 is involved in brain development. You have rapid brain development if you are not Homo sapien. Yikes, what are you saying? We have a very prolonged juvenile stage. Our brain development has been purposely slowed down, well, purposely in the purpose of evolution, to give us a longer window that we have flexible brains where we can learn. And that pretty much has made culture, higher culture, possible. SIRGAP2, it's involved in controlling the migration of neurons as the neural layers of the brain are established. It's also involved in the morphology of the stem structures. If you look at the dendrites here, if you could zoom down, you'll see it's composed of little heads and stems. In non-human mammals, you don't have as many, and they have little short, stubby head stem structures. In the mutant form, that's us, you have more of them, and they have more elongated necks. This is called the neck. So what does all that mean? How does this work? The mutant form combines with the original form and it becomes insoluble. It's like an amyloid plaque or something. It precipitates out and is non-functional. This mutant form depresses or represses, interferes with the function of the original SIRGAP2. And the result is that we have slowed down our intellectual and brain development, which on the surface we go, whoa, don't want that, I'm already having enough trouble. But it's allowed us to develop culture. Increased capacity for cognitive brain function correlates with the beginning of the neocortex, expansion of the hominid line. This is where the different mutations have occurred. Notice how they're correlated with the different fossil forms of the hominids. The first time it duplicated, it was before Lucy. Lucy was 3.2. Then, around the time of Homo habilis, 2.4 million years ago, that was the second duplication. And then the third duplication was here, where you get all of these forms, and antecessor, Heidelbergensis, fire. We first began to deliberately play with fire way back here, almost a million years ago, while we were still pretty stupid. We were playing with matches. When did you start playing with matches? Probably when you were pretty stupid. So, of course it's not one gene. But this gene has been expressed in mice. And when you look at brain development, that's how we know its effect on the dendrites. It actually had that effect. Didn't make the mice go, I'm not going in that mouse trap, get that cheese. You go get it. It takes a family of mutations, but we seem to have identified some that are involved in the early changes that happened that ended up making us who we are. Neonatalism. That's this juvenile tendency. You know, you see it in pets, right? Wolves became bulldogs. The snout. It turns out that the genes involved in prolonged juvenile stage, dogs are really forever puppies, those genes are physically linked with face morphology. And so as you select for nice dog, you're also selecting for flat-faced dog. They begin to get all, we have selected ourselves, our big protruding lower mandibles in the apes. We selected, as we were selecting for a kinder, gentler, we were also changing inadvertently the structure of our very face. Our teeth don't fit right now. We're still rapidly changing. We have selected, like we did, you see it in pigs, you see it in dogs, you see it in lots of domestic animals, where the domestication ends up in the loss of the protruding snout, just because these genes in mammals are closely linked. It's just a byproduct. We did that to ourselves. Here you see the juvenile. So we have prolonged the juvenile state, because this chap looks like us. As a chimp grows up, he begins to look like Rufus here. Oh, this guy is Rufus. I'll tell you a little story and then we'll leave. I was my parents' oldest child, and I lived in a small textile town. We lived beside these old retired people, and they would go fishing. They had this gravel driveway, and the guy's name was Rufus Mountford. And he would go fishing early in the morning, and I was still at the feeding stage where every 2 hours you go in and feed. My mother would get up and feed me. And one morning, she got up, it was like 4 o'clock in the morning, do my 4 o'clock feeding, and she got in bed. She said to my father, his name was Byron, she said, Byron, the Rufus Mountfords have gone. And he sat up in bed. The roof of his mouth is gone. He went running in there, took me from the crib and feel like that. No, the Rufus Mountfords are gone fishing next door. Okay. We'll see you next Thursday.